Hello everyone and welcome to my presentation. My name is Jeroen Gilis. I am a PhD student at Ghent University at the Statomics group that is headed by Professor Lieven Clement. Today I will be presenting a novel R software package called satuRn for performing differential transcript usage analyses. The concept of differential transcript usage is closely related to the concept of alternative splicing. Due to alternative splicing, multiple isoforms can be created from a single genomic locus. And these isoforms may have completely different functions. Now, when we perform a classical differential gene expression analysis, for instance with HR or DE-seq2, we focus only on the total expression output of a gene, without recognizing the fact that the different isoforms within that gene could be expressed in completely different proportions. Therefore, differential transcript usage analyses, or DTU analyses, provide an extra layer of information on the same experiment. Over the past few years, several DTU tools have already been developed, but we argue that none of these tools are optimal, in the sense that there are no tools that satisfy all of the following criteria: a high performance, a good scalability, an accurate control of the false discovery rate, the ability to model multi-factor experimental designs, and robustness against real-life proportions of zero counts in single-cell data. Therefore, it was our objective to develop such a method. To describe the statistical framework that is used by satuRn, we will use the notation Y subscript GTI to denote the expression of a transcript T that belongs to a gene G in sample or cell I. The total expression of gene G in sample I can then be expressed as the sum of the expression values of each of the individual transcripts within that gene. Next, we can very easily estimate the usage of transcript T in sample I by dividing the expression value of the transcript with the total expression value of all transcripts in the corresponding gene. Next, we can model the usage of transcript T by adopting a quasi-binomial generalized linear model, or GLM. In this notation, pi subscript GTI corresponds to the estimated usage of transcript T in sample I. Pi GTI is linked with a logic link function to a covariate pattern X, which allows for having different transcript usage estimates in different groups of samples or in different cell types. Finally, the distinguishing feature of the quasi-binomial GLM as compared to a logistic regression model is a function that describes the variance. In addition to the canonical variance function of the logistic model, we have the factor phi subscript GT, which is a transcript-specific overdispersion parameter that describes additional variance in the data with respect to the binomial variance. On the left-hand side of this slide, we display the performance of satuRn with respect to 5 other DTU tools from the literature on a set of 4 benchmarks datasets. The top 2 panels are benchmarks based on a real bulk RNA-Seq dataset that were filtered stringently with the filtering criterion implemented in DRIM-seq. We assess DTU in a 2-group comparison of 5 versus 5 and 50 versus 50 samples, respectively. The bottom 2 panels are benchmarks based on a real single-cell dataset that were filtered leniently with the filtering criterion from HR. Again, we assess DTU in a 2-group comparison of 20 versus 20 and 50 versus 50 cells, respectively. These figures display FDR-TPR curves, meaning we have a measure for specificity on the x-axis and a measure for sensitivity on the y-axis. Here we clearly see that the performance of satuRn, which is shown in black, is on par with the performances of the best tools from the literature. The 3 circles on each curve represent working points when the FDR level is set at a nominal level of 1%, 5%, and 10%, respectively. The circles are filled if the empirical or observed FDR is equal or below the imposed FDR threshold. From these points, we can see that satuRn provides stringent control of the FDR in all 4 benchmarks, whereas other DTU methods such as double XPseq become increasingly liberal with increasing sample sizes. On the right-hand side of the slide, we demonstrate the scalability of the different DTU tools. On the top 2 panels, we display the scalability of each method with respect to the number of cells in the dataset, while keeping the number of transcripts in the data fixed. Here we see that DRIM-seq and especially DEX-seq scale poorly with respect to the number of cells in the data, jeopardizing their use for analyzing large single-cell datasets. On the bottom 2 panels, we display the scalability with respect to the number of transcripts in the dataset, while keeping the number of cells in the data fixed. Here, we observe that Bandits scales particularly poorly to the number of transcripts in the dataset. In contrast, satuRn scaled seamlessly to datasets with a large number of cells and a large number of transcripts. We then used satuRn in a real single-cell case study to identify differentially used transcripts between the different cell types of the mouse neocortex. In this slide, I have focused on a single exemplary gene. In a canonical analysis with HR, we found no evidence for differential gene expression of the P2RX4 gene between these 2 mouse neocortex cell types. However, when we looked at DTU with satuRn, we did observe large differences in the usage of the respective isoforms of the P2RX4 gene. More specifically, we observed that the isoform in the top panel is clearly the most abundant isoform in the blue cell type, with an average estimated isoform usage of approximately 75%. By contrast, the isoform in the bottom panel is the dominant isoform in the red cell type, with an estimated average isoform usage of approximately 60%. Interestingly, the isoform in the top panel is protein coding, while the isoform at the bottom is not. As such, this discrepancy in the isoform usage profiles between these 2 cell types may reflect a biologically relevant discrimination between both cell types. As a small wrap-up, we have developed satuRn, which is a novel method for performing differential transcript usage analyses. The principal features of satuRn are its high performance, its great scalability to large datasets, its ability to control the false discovery rate close to the desired level, the fact that it allows for modeling multi-factor experimental designs, and its ability to handle real-life proportions of zero counts. The statistical framework of satuRn relies on a quasi-binomial generalized linear model. In a case study, we have shown one example of how satuRn was able to detect differences in transcript usage between cell types that are likely to be biologically relevant. And finally, satuRn will soon be published on bioRxiv and available as an R package from GitHub. Thank you for your attention.